Hey, first and third hour, this is Miss P. Sorry I can't be there today. I am out on a field trip with Bio Club. So anyway, we're just going to go over very briefly replication right now, and then we'll do transcription and translation tomorrow together when I'm back. So, why can I not advance the slide? There we go. There are 8 understandings for this unit, but only a few for replication. So that's where we're going to focus today, just these 5 pages in your book. So the first thing that you need to know is helicase, which we know is an enzyme because it ends in ASE, right? Breaks our double helix. So we know what our double helix looks like now because we did our structure of DNA a few days ago. And we know that it breaks the hydrogen bonds. So if we look here, here's the structure of DNA, right? And you can see the nucleotides here with the hydrogen or nitrogen bases right here, 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 so on and so forth. And then there is a hydrogen bond between the nitrogen bases. Remember, A pairs with T and G pairs with C. And what happens is helicase doesn't really look like this. It looks more like this, but it's drawn to essentially look like a zipper. And it unzips or busts the hydrogen bonds as it slides down. And as it slides down, then the A's, T's, C's, and G's are available to be copied. Now this needs energy, right? Because you can't just do that willy-nilly. And let's see if I can go ahead and write. So here's what you need to know. Do you remember that DNA has 3 prime ends and 5 prime ends, right? So if I can get my pen maybe over here. This is, oh, maybe I can't write. This is our 3 prime end over here, which would mean that then if I follow this all the way down over here, right here is the 5 prime end of that same thing. And then the same thing here, this, well, I can do this, 3, 5, sorry for the clicks. Then, remember, the opposite is true for DNA, the other side. So this is 5 then, and then this is 3. So DNA is always read, the old DNA is always read from 3 to 5. So this one will be read this way, and this one will be read this way. So let's look at that a little and further. So when we go to make another strand of DNA, we know that it's read one way or the other, but we actually need to physically make the other strand. And so we use the template of the old DNA to make the additional new DNA strand. And what the enzyme that we use is called DNA polymerase, ASE, right? And we know that it makes DNA because it's called DNA polymerase. If we were making RNA, it would be called RNA polymerase. And it does a condensation reaction, so you should be fully aware of what that means. So here is my helicase unzipping. This is called a replication fork. They are together here at this point. Helicase hasn't broken them yet, the hydrogen bonds, and now all of a sudden those bonds are broken. So remember, this was the 3 prime end, down here was the 5, over here is the 5, over here then is the 3. And remember, that's just based on the sugar that's facing closest to the side. So here is a yellow blob representing DNA polymerase. And so it sits down, sees an A, puts a T, sees a C, puts a G, so on and so forth all the way down. Now as helicase unzips, 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 this DNA polymerase can keep going, right? So this whole strand is made in one piece because as helicase unzips, DNA polymerase can keep following it, and it's called one giant strand, also known as the leading strand because it's one big strand. Now if this is the 3 prime of the old, then that means that this needs to be 5 and would end in 3, right? Because they're always opposite of one another. This will run this way, this one will run this way, right? Now on the other side, because DNA has to be read this direction, it can't be made actually in one big giant strand. It needs to be made in little segments. So what happens is DNA polymerase makes this little bit, and then jumps back physically, like leapfrogs back, and makes this little bit. And then as this unzips more, it would make another little bit, and then another little bit, and then another little bit. And then eventually they'd all get sealed together. So they'd still be one big strand on the end. But it is important to know that DNA polymerase is moving to the right on this strand and to the left on this strand. Now, ladies and gentlemen, that's not always the case. If we would have put 5' up here and 3' down here, they would have been the other direction. But please do know that they're always moving in the opposite direction. All right, so how does it bind? Well, you remember it makes these lovely little bonds called phosphodiester bonds. They're covalent bonds. And if the old DNA strand is read from 3 to 5, then the new DNA strand is made from 5 to 3. So I'd suggest remembering one of those and then knowing that the other strand is opposite, however you want to remember it. So it says DNA polymerase always moves from 5 to 3. That is in fact correct. If that's the easiest way to remember it, that's fine. So when we make a new strand, it starts always with 5 and always ends with 3 whenever we make a new strand. So we start with 5 and we would end with 3', which would be this sugar down here, right? Please remember that A's always pairs with T, G always pairs with C, that there are 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. When A bonds with T and G bonds with C, that's called complementary base pairing because they are pairing with their associated base. So that's the first thing that happens. The second thing that happens is that eventually you get 2 identical DNA strands. So base pairing happens, and if you'll notice, if you look at this segment here versus this segment here, I'll move me up here for a second. Whee! You can see that they match, right? They're complementary, they're identical. So eventually we get these 2 identical strands. And so what we call this is semi-conservative because the blue is old, but the green is new. So it's semi-conserved, half old, half new on both strands. Okay, I'm going to put these links on Schoology because I can't play them on the video because you won't be able to hear the audio, so it won't do you any good. You won't be able to use your iPads for these videos, so you will have to go to the back and use one of the computers back there, or if one of you have a computer, you can use that as well. But I will put the links on. Essentially, it's this process called PCR, which stands for polymerase chain reaction. And this is what happens. When we want to copy DNA, we always want to just copy a specific section. So we heat up the enzyme, and what happens is all those hydrogen bonds break apart between the 2 strands. So they split apart. And then we put on a primer. It says, hey, start here. And we put on this primer that says, hey, start here. And then DNA replication happens just like what we just talked about. And so we end up using a specific type of polymerase called Taq that's really resistant to heat because we have to warm it up really, really warm and cool it down and warm it up again. And if we use the DNA or the DNA polymerase that's in our body, it would break apart, it would denature. So we use these Taq polymerase that we find in actually bacteria that live in hot springs. So it works really well. So we make a copy of it, and now all of a sudden we make a copy of this one and we make a copy of this one. So now we have 2 complete copies. And then what we do is we repeat the whole thing. We heat it to separate it, we cool it down to attach the primers, and then we extend the nucleotide sequence. And then we do it again and again and again and again. And anywhere, when we do PCR, and we will, we do about 30 cycles. So we amplify the DNA of one copy to, quite a ton. So anyway, that's the process of PCR. Please do watch at least one of those animations to make sure that you know it. And the last thing that I want to talk to you about is how did we figure out what semi-conservative was for DNA? So here's what happened. Milsel and Stahl did something very simple. They took nitrogen 14 and nitrogen 15. And again, I'm going to put a link on Schoology because I think it would be really helpful for you to watch this. And I don't think I can change my pen color. Oh, I can. Look at that. Okay, so we have this DNA that was all one weight, right? And we put it in a base that is the different weight. And so what we ended up seeing is when that DNA went to replicate, so let's say our nitrogen 14 is our blue, the only nucleotides that it had floating around in this little petri dish were not for N14, but were for N15. So now all of a sudden the nucleotides that are on this side are all N15 instead of N14. That would make these DNA strands heavier than this DNA strand, right? Does that make sense? And then what they did is they did another petri dish, but instead of N15 now they had N14 set in there. And now these strands separated again. So we actually had two, I'm just going to draw them like this now. We had two green and we had two blue, totally separate from one another, right? Because these separated right down the middle. And now because they had N14, the other sides bonded with N14. Now if you took this DNA and spun it around really, really fast, you'd have a band of medium density and a band of really, really light density. And so through that process, we actually figured out, or we didn't, they did, that DNA was semi-conservative. And so that was a really cool finding, that when you weight nitrogen with different weights, you can actually see a different banding pattern in the test tube. And I think it's really important for you to watch this animation because I think it will help make sense. So here's the heavy DNA, here's the light DNA, here's the combination of the heavy and light as I just talked about. And this does a really nice job showing you how old DNA, old and new, new, old and new, and what the bands would look like accordingly. But this animation walks you through that. So take some time at the end of class, watch the two animations, and work on pages or questions 1 through 13 in your white packet that Ms. Dubé should be giving out to you. That will be stuff that you want to kind of get wrapped up before we start tomorrow, because tomorrow is kind of a new topic in itself. I'll go over any questions tomorrow, but hopefully that started to make sense. Have a good day.